What are tumor specific proteins? Tumor specific proteins means these proteins are expressed only by the tumor cells. Example, an oncogene is translocated and fused to an active promoter of another gene. A fusion gene formed and this fusion gene results in fusion protein. Constant active production of this fusion protein and development of malignant clone. Philadelphia chromosome in chronic myeloid leukemia, translocation of 9 and 22nd chromosome. The Q arm of the 34 and Q arm of the 11 chromosome, BCR-ABL translocation also in chronic myeloid leukemia. T821 in acute non-lymphatic leukemia, T1517 translocation. T stands for translocation. The numbers there are the chromosomes that are translocated in APL, that is acute plasma cell leukemia. Other hematological malignancies also, tumor specific proteins are seen. We will see a little about chronic myeloid leukemia and the Philadelphia chromosome in our next slide. What is Philadelphia chromosome? This is in fact the first tumor marker or a chromosomal abnormality that is related to malignancy. In 1960, Philadelphia chromosome is detected in a patient of chronic myeloid leukemia. Since then, it has become a marker of CML. It is an abnormal chromosome due to translocation. Translocation means a part of one chromosome is cut off and attached to part of another chromosome which is also scissored off, and the new fusion chromosome will be the abnormal chromosome. The translocated chromosome is also called the Philadelphia chromosome or PH chromosome, a short in its structure and size, which we can see in the next slide. There is a BCR-ABL fusion gene, the BCR gene of the 9th chromosome and the ABL gene of the 22nd chromosome are fused to produce a BCR-ABL fusion gene. This fusion gene triggers chronic myeloid leukemia. This takes place in a single bone marrow cell. That single cell wherein this Philadelphia chromosome is formed due to translocation multiplies and it clones, becoming more and more, and ultimately the whole bone marrow and blood is invaded by chronic myeloid leukemia cells, creating fusion proteins. This is detected by a test called FISH or FISH test, fluorescent in situ hybridoma or hybridization technique. Fluorescent in situ hybridization technique, this is the fluorescent in situ hybridization, and you can see the fluorescent materials there, and they are the Philadelphia chromosomes. This is the chromosome 9 and this is the chromosome 22, and this part of chromosome 9 is truncated and is attached to this part of the chromosome 22, and the fusion BCR-ABL fusion gene occurs, and that is what is the Philadelphia chromosome. Philadelphia chromosome, if it occurs in acute lymphatic leukemia, it suggests poor prognosis. You can diagrammatically see here the normal chromosomes. These are the normal chromosomes, the 9th chromosome and the 22nd chromosome. So the 9th chromosome is truncated here and a small bit of this is taken, and the 22nd chromosome is cut off here, and that small bit of the 22nd chromosome, this is left out, and this bit and this small bit, these two to join to form what is called the abnormal Philadelphia chromosome, and the 9th chromosome is completed by addition of this small bit of the 22nd one, and this is the changed 9th chromosome, and this is the changed 22nd chromosome, which is also called the Philadelphia chromosome, which we have discussed. It is diagnostic of chronic myeloid leukemia, and its presence in acute lymphatic leukemia suggests poor prognosis.